Hello and welcome to Scientists of the Spot on Cardiovascular Research Onlife and the Scientists of Tomorrow. I'm Niels Voigt from Göttingen University and I have with me today Professor John Lederer from the University of Maryland, from Baltimore. So John, welcome and thank you for taking this interview. The Scientists of Tomorrow are very keen to listen to your career and to the advices you may have for us. So the first question is related to your science. You made many great discoveries in cardiac biology. So for you, what do you think are the most important ones, the highlights?
Well, I think the first thing I'd say is I didn't make all of these discoveries. I worked with a group of people who were students, postdocs, visiting faculty members, an amazing group. And together we talked and we're trying to understand various aspects of whatever we were working on, the heart, mainly in calcium signaling to start with. I would say the most significant single discovery was calcium sparks. And calcium sparks, when discovered, was with Mark Cannell and Peace Chang. And when we first discovered calcium sparks, we had to rule out there being an artifact. We, in fact, didn't know what they were. We didn't know what to call them. We, and this was mainly Mark, rebuilt our confocal microscope to try to get rid of that noise that turned out to be sparks. After the fact, they were perfectly obvious and large. You didn't need a confocal microscope to see them. But until then, that's how it was. So I would say the largest, most important discovery was the discovery of calcium sparks in heart. And I stress heart because, of course, in heart, it led to a lot of other things. But we also discovered calcium sparks in vascular smooth muscle. Completely different result. It produced a surprising result. It doesn't produce contraction, the way it does in heart. It produces relaxation. Calcium activates potassium channels. And in skeletal muscle, we discovered sparks. We were thinking about the same thing now, once started down that path. And so the smooth muscle sparks, that was done with Mark Nelson. And some of the other guys too. And the sparks in skeletal muscle, that was done with Martin Schneider and members of his lab and some of the other guys. And we've seen sparks in lots of different other places. So the prominent, discovery was calcium sparks, and we've been discovering things about them ever since, as have many other people worldwide. So I would say that's probably number one. But there are a lot of others.
So probably related to this question, you worked with myocardial calcium homeostasis for many decades. What do you think the field is developing in the future?
I think the field is continuing in the same pattern of discovery, which is not surprising. The pattern that we have been involved with has been finding a phenomenon and seeing how it works. And usually what we see is different than what we expected. And the delightful surprise is that we start with one idea and discover we were wrong about it. We find something new, we're wrong a little bit, and it progresses through an iterative pathway where we're wrong and right and wrong and right. And so maybe the biggest discovery along this path, for calcium in the future in heart really applies more generally to all discoveries, which is it's good, not necessarily something you desire, but it's good to be wrong. Find out that the things you think are true aren't quite true the way you think. And by exploring those aspects, we learn more.
So from your perspective, how do you define great science, and what is the role of technical evolution in the scientific process?
Well, let me start with technical evolution. Like many other people who sort of dabble in the engineering aspect of measuring things, the technical developments along the way have been huge. They're terribly important, they're fun, and they enable us to do things that we thought maybe not possible before. Just take, for example, super resolution. We have been involved now in some project with all of the different forms of so-called super resolution. We certainly know well about Stefan Hell, and that's thoroughly supported by people doing STED, super resolution microscopy here at Göttingen, but of course we're doing it in Baltimore and people are doing it around the world. That's one kind of technology that changes how well you see things, how fast you can see things, and so on. And so, of course, STORM and PALM are other good examples. But there are other things that are really helpful. For example, I don't know if you are aware of it, but there is an imaging flow cytometer that works at high resolution. And so you can see objects and count them. And that's technology at work. It helps you learn more. And we can go down the list. It is the new technology helps us fulfill the dream experiments that we've had. And if we work hard and are careful, maybe we'll figure things out, you know, one thing or another.
From your MD perspective, how did your discoveries, or how will they influence the treatment of patients?
Well, huge. But you know, maybe everyone feels that way. Treatment of patients, new treatments, better treatments, the treatment of things we don't know about, all depends on careful research. And what research is about is discovery in this iterative cycle of finding something out, finding that what you thought was true was wrong, figuring out why, and doing it again. And so it's pushing back a kind of intellectual frontier all the time. It's exciting and people love to do it. It's like being a kid in a candy store, finding new things that are tasty and sweet, and you consume them. So how does it help the practice of medicine? Well, medicine is about trying to treat disease. Most disease that's understood can be pretty well treated. You know, if you have an antibiotic, you can treat a disease. If you have an anti-cancer agent, you can treat the cancer. But after a while, they don't work as well, or maybe you have a variant of the disease, you need to do more. Our work on sparks and mechanical translation transduction led to the discovery of XROS signaling, a physiologic reactive oxygen signal that modifies calcium signaling and other things. Well, that led to our better understanding of microtubules and their interaction with proteins and how they work and how they cross-link. We have insight into hypertrophic cardiomyopathy. We have insight into cancer migration with other colleagues who are working on how cancer invades as a metastatic agent. We have understanding about traumatic brain injury and how the NOX2 proteins on the surface of brain cells are affected when the brain is shaken and the microtubules pull. And it goes on and on and on. And this is from, if you like, a derivative of discovery. You know, so I think the long-term benefit is huge, but I don't know the path, you know, it will take us on, or what we need to follow to get there.
John, you mentored many famous scientists who are now leaders in the field. So what is your trick, or what is your tip? Do you have any advices you can give to our audience? What would you recommend them to the young basic scientists?
Well, personally, they get the credit. I mean, you know, these are individuals who came to the lab or we met and liked to work, and somehow we found things that were interesting. I would say probably the most important thing is to teach the people you are trying to mentor to have fun. This is a project or set of projects that you're going to work on that will take you places that are unknown, you hope. If you go back to what you know, you haven't made too much. So be bold. Don't worry about being wrong, because you will be. So it's really somehow trying to lose the fear of failure and being willing to explore with moderation, you know, read the literature, do the hard work, but it's about exploration. So, you know, each person does it him or herself. It's not, I'm not doing it. They are becoming the great people they are, not anything else. And I can think of only a few people in the lab who really, you know, didn't find that exploration as exciting as the others. But almost everyone else has come to the lab, and, you know, people don't want to leave, but, you know, they have to make a living. They have to start their own labs, and they have. And we've been lucky for it.
Thank you very much for giving us this interview and coming all the way from Baltimore to attend the Göttingen Channels meeting in Germany. And on behalf of Cardiovascular, Onlife and the scientists of tomorrow, I would like to thank the audience for watching to this podcast.